Hi everyone, I'm Madeline and I am presenting on the determination of gender, genes versus hormones. So first I'll do a quick little overview of male and female sex organs. So the male and female sex organs consist of three sets of structures, the gonads, the internal genitalia, and the external genitalia. Gonads are the organs that produce the gametes, or the eggs and sperm that unite to form new individuals. The male gonads are the testes, which produce the sperm, and the female gonads are the ovaries, which produce eggs or the ova. The undifferentiated gonadal cells destined to produce eggs and sperm are called the germ cells. The internal genitalia consist of accessory glands and ducts that connect the gonads with the outside environment. And the external genitalia include all external reproductive structures. So now I'll go a little bit of a chromosome overview here. Sexual development is programmed in the human genome. So each nucleated cell of the body, except for eggs and sperm, contains 46 chromosomes. This set of chromosomes is called the diploid number because the chromosomes occur in pairs, 22 matched pairs of autosomes plus one pair of sex chromosomes, which is seen in the top figure to the right. The amount of DNA in a diploid cell is written as 2n, indicating duplicated chromosomes. So the 22 pairs of autosomal chromosomes in our cells direct development of the human body form and of our varying characteristics such as our hair color, eye color, our blood type. So the two sex chromosomes are designated as either X or Y, contain the genes that direct the development of our internal and external sex organs. So you can kind of see the X and the Y chromosomes in that bottom right figure there. The X chromosome is much larger than the Y chromosome and includes many genes that are missing actually from the Y chromosome. So eggs and sperm are the haploid cells with 23 chromosomes. One from each of the 22 matched pairs, plus one sex chromosome. When egg and sperm unite, the resulting zygote then contains a unique set of these 46 chromosomes, with one chromosome of each matched pair coming from the mother and the other from the father. So the sex chromosomes that a person inherits is what determines our genetic sex makeup. So genetic females are XX, genetic males are XY. Females inherit one X chromosome from each parent, and then males inherit a Y chromosome from the father and an X chromosome from the mother. The Y chromosome is essential for development of the male reproductive organs. If sex chromosomes are abnormally distributed at fertilization, the presence or absence of a Y chromosome then determines whether development proceeds along male or female lines. So the presence of a Y chromosome means the embryo will become male, even if the zygote has multiple X chromosomes. So if they're XXY, the zygote will still become a male. A zygote that inherits only a Y chromosome, so no X chromosome, will not be able to survive and will die because the larger X chromosome contains essential genes that would be missing from that Y chromosome. In the absence of a Y chromosome, an embryo will develop into a female. So for this reason, a zygote that gets only one X chromosome would end up having what we know as Turner syndrome, but they still will develop into a female. Two X chromosomes are needed for normal female reproductive function. However, once the ovaries develop into a female fetus, one X chromosome in each cell of her body is inactivated and condenses into a clump of these nuclear chromatin, which is known as the Barr body. The selection of the X chromosome that becomes inactive during her development is random. Some cells will have an active maternal X chromosome and others might have an active paternal X chromosome. So because inactivation occurs early in their development, before the cell division is complete, all the cells of a given tissue will usually have the same active X chromosome though, whether it's maternal or paternal. So the sex of an early embryo is difficult to determine because reproductive structures don't begin to differentiate until about the 7th week of development. Before differentiation, the embryonic tissues are considered bipotential because they cannot be identified as male or female. The bipotential gonad has an outer cortex and an inner medulla. Under the influence of appropriate developmental signals, the medulla will turn into the testes. In the absence of that signal, the cortex will differentiate into the ovarian tissue. The bipotential internal genitalia consist of two pairs of accessory ducts, the Wolffian, which are derived from the embryonic kidney, and the Mullerian ducts. As development, proceeds along either male or female paths, one pair of ducts develops while the other degenerates. The bipotential external genitalia consist of the genital tubercle, urethral folds, urethral groove, and the labioscrotal swellings. These structures then differentiate into the male and female reproductive structures as their development progresses. So what directs some signal cell zygotes to become males and others to become females is sex determination based on the presence or absence of that sex-determining region of the Y chromosome. So we call this the SRY gene. In the presence of a functional SRY gene, the bipotential gonads develop into the testes. In the absence of the SRY gene, and under direction of multiple female-specific genes, the gonads develop into the ovaries. The figure on the right shows the SRY gene effects on male embryonic development. So in the male embryonic development, the SRY gene produces the protein testes determining factor, or TDF, that binds to their DNA and activates additional genes, which would include SOX9, Wilms tumor protein, and steroidogenic factor. The protein products of these and other genes direct development of the gonadal medulla into the testes. So testicular development actually doesn't require male sex hormones like testosterone, as most of us would think it does. The developing embryo cannot secrete testosterone, in fact, until after the gonads differentiate into the testes. Once the testes differentiate, they begin to secrete three hormones that influence development of the male internal and external genitalia. And these are testicular Sartori cells, which will secrete the glycoprotein anti-Mullerian hormone, which is the AMH. Testicular interstitial cells secrete the androgen, so testosterone, and its derivative dihydrotestosterone, or DHT. Testosterone and DHT are the dominant steroid hormones in the males, and both bind to the same androgen receptor, but the two ligands elicit different responses. So in the developing fetus, anti-Mullerian hormone, or AMH, causes the embryonic Mullerian ducts to regress. And testosterone converts the Wolffian ducts into the male accessory structures, of which are the epididymis, the vas deferens, seminal vesicle. Later in fetal development, testosterone controls migration of the testes from the abdomen down into the scrotal sac. And the remaining male sex characteristics, such as the differentiation of the external genitalia, is controlled mostly by DHT. The importance of DHT in male development really has came into light when they were studying individuals with pseudo-hermaphroditism. So these men inherit a defective gene, which is the 5A reductase, and it's the enzyme that catalyzes the conversion of testosterone to DHT. So despite normal testosterone secretion, these men have inadequate levels of DHT. And as a result, the male external genitalia and prostate gland fail to develop fully during their fetal development. So at birth, these infants have a female appearance, and they end up being raised that way. But then at puberty, the testes begin to secrete testosterone and cause masculinization. So by studying this 5A alpha reductase defect in these individuals, scientists have been able to separate the effects of testosterone from DHT. And this figure on the right shows that synthesis pathways for some of these steroid hormones. So now we'll go over the female embryonic development. So in female embryos, which have the no SRY gene, the cortex of the bipotential gonad develops into ovarian tissue. Research has actually indicated that females' development is a little bit more complex than we had originally thought, with multiple genes being required for the development of functioning ovaries. And in the absence of this testicular AMH, the Mullerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. The fallopian tubes are also called the uterine tubes or the oviducts. So without testosterone, the Wolffian ducts degenerate, And without DHT, the external genitalia will take on the female characteristics. So there's a little assignment to test yourself about all the things I just went over.